Cells are generally very small and are invisible without a microscope. Eukaryotic cells vary between 10 and 100 micrometers in diameter, while prokaryotic cells range from 0.1 to 5.0 micrometers in diameter. The invention of the light microscope in the early 19th century made the study of tissues and cell structure possible. This allowed the development of the cell theory, as well as accumulation of a large amount of information on the similarities and differences among cells from a huge number of organisms. Through innovations in the field of microscopy, scientists have been able to develop a better understanding of cell types and cellular structures. Cells were originally categorized as two main types, prokaryotes and eukaryotes. Furthermore, it was Carl Woese and others that classified all life on Earth into three domains: bacteria, which are prokaryotic; archaea, which are also considered prokaryotic; eukaryota, which are obviously, as the name suggests, eukaryotes. The main differences between prokaryotic and eukaryotic cells are: eukaryotic cells have a separate membrane-enclosed nucleus, whereas the DNA of prokaryotes is freely floating in the cytoplasm. Eukaryotic cells have a complex system of membrane-bound organelles that divides the cell into numerous enclosed regions known as compartmentalization, but this will be later focused on and expanded more on in a separate video about eukaryotes. Lastly, prokaryotes do not have any membrane-bound organelles. Thus, in this video by BMAT Edu, we will be going over prokaryotes and its structures. To learn more about cell biology topic B1 from BMAT, check out our other video about eukaryotes linked in the description. So, prokaryotes. Prokaryotes are considered to be the earliest and most primitive type of cell, originating some 3.5 billion years ago. They include bacteria and archaea, and these unicellular organisms belong to different domains due to differences in the cell wall composition and RNA. The domain of prokaryotic shows an enormous variety of species. They are found everywhere, including the most inhospitable places you can think of, from boiling wells to deep mine shafts to the ocean floor. Additionally, they have the simplest system that allows all processes of life to be carried out. They are simple unicellular organisms with no internal compartmentalization, no nucleus, and no membrane-bound organelles. Hence, all metabolic processes thus occur within the cytoplasm. Talking about the cellular structures of prokaryotes, some cellular structures that you would need to know and its functions are obviously the cell wall, which encloses the cell, protecting it and helping to maintain its shape, prevents the cell from also bursting in a hypotonic dilute media. There is plasma membrane, which you can see here, surrounds the cell, controlling the movement of substances in and out of the cell. There's a cytoplasm, which is a medium that fills the cell and is the site of all metabolic reactions. There's pili, which in singular form is pilus. It's protein filaments on the cell wall that help in cell adhesion and in transferring of DNA between two cells. There's flagella, which in singular form is flagellum. It is much longer than pili. These are responsible for the locomotion of the organism. Their whip-like movement propels the cell along. There's 70S ribosomes, which are the sites of protein synthesis. Most importantly, there's the nucleoid region, which includes naked DNA, that is DNA not associated with proteins known as histones. It controls all the activities of the cell wall, as well as it functions to take care of the reproduction of the organism. And lastly, there's plasmids, which are small circles of DNA that carry a few genes. Often these genes give the cell antibiotic resistance and are used in creating genetically modified bacteria. Additionally, prokaryotes reproduce by binary fission. Basically, they split in half, as shown in the diagram, to produce two genetically identical cells asexually. The steps are as follows. The chromosome is replicated semi-conservatively, beginning at the point of origin. Beginning with the point of origin, the two copies of DNA move to opposite ends of the cell elongates, and the plasma membrane grows inward and pinches off to form two separate genetically identical cells. that will be all for today. Again, don't forget to check out our other video on eukaryotes and subscribe to our channel. In the description, links to worksheets and answer keys related to this topic are also available. And do take a look at our website as well if you would like to learn more about us, learn more about the content that we make and when we post our stuff. And even if you want to contact us, let us know in the comment section below and in our website. And with that, thank you for watching. All the best with your preparation.